Hi everyone, welcome to today's webinar on accelerating ATMP development from lab to GMP. My name is Kate O'Dwyer and I am one of the process development engineers and also CMC specialists at APC and VLE, and I am very delighted to be taking you through the presentation today. So straight into our presentation, I'm going to go firstly through the product. So this product was an oncolytic virus. Oncolytic viruses are quite big. They're about 300 nanometers in size, and this particular drug was used as an immunotherapy for patients with advanced malignancies. The goal of this drug was to kill infected cancer cells through oncolysis and ignore any unaffected cells via tumor targeting. So this drug would have been injected into tumors. You can see here our virus goes through healthy cells and doesn't damage them, but when it hits a tumor cell, viral replication occurs, and then after that, immunogenic cell death or oncolysis occurs then when our tumor cells get lysed. The cell line, so this was an adherent cell line. The product was made using HeLa cells, and it was an intracellular product. So our virus would have been replicated inside of the HeLa cells, meaning that we had to lyse them later on in the process. And one thing to shout out for those of you that don't know is that HeLa cells were a cell line that was taken from the tumor of Henrietta Lacks, who was a lady in, I believe, the 50s. Who unfortunately died of cancer, but her cells were used to create the immortal HeLa cell line, which is one of the most used cells for drug production in today's world. So I always think that it's really important to call out where the word HeLa comes from, or where these cells come from. And then the process was an adherent cell line process, as I said, using the HeLa cells, but this all would have been on kind of flatware. So you can see our C train is on type of cell factories or cell stacks into a roller bottle process and then onto kind of a 40 roller bottle process. The harvest volume was 8 liters, so quite a small harvest volume. And the productivity was about 140 plaque forming units per cell. The phase that we were at in this, I think, is really important to get the context of why we were doing this project at APC and VLE. So the drug at the time was in phase 2 clinical trials. When the process was first developed, it wasn't developed with the end in mind, for example. And the impurity profile that we'll talk about later in the downstream, so our host cell DNA, our host cell protein, and kind of our residuals, like any enzymatic digestion agents that we were using, the levels of these didn't meet what was required for our phase 3 clinical trials, because the further we go on, the more regulatory rigor and more tightness that we need around our controls. And as well, our upstream wasn't scalable to treat the patient demand or to kind of have a robust upstream. It was very manually intensive. And you can see there only an 8-liter harvest volume and quite a small surface area. So if we were going to scale this to the patient demand, we would need a warehouse full of those ORB40 roller bottle units. So the project goals was to create a scalable GMP suitable process, increase the titer, reduce impurities, control our critical process parameters, and ensure that our CQAs were comparable or improved upon. So we have to make sure that because we were in the middle of clinical trials, that whatever we were doing to change the drug, or whatever we were doing to change the process rather, kept the CQAs the same from a statistical point of view. So how were we going to do this? Through our CMC strategy, so we use CMC maps and CMC milestones which are infused into our programs at kickoff. And the client, APC, and VLE are all responsible for their own milestones, but it's a good way to keep everybody on track in terms of when we know that for this date we need to have a certain work package done with a report or some type of deliverable on the end of the project because of this very important clinical phase or filing that the client's going to do. We used our online knowledge management platform, iAchieve, as our one source of truth. So getting in our client data, doing our process development work, and then scaling that to our GMP colleagues over in VLE, having everything in the same place was really important and very, very useful to the team. We used accelerated process development. So we started with the end in mind, looking at manufacturability, our operators in VLE kind of being on the journey with us, and then the procurement of our raw materials and our single-use technologies. So ensuring that everything that we used or planned on using could be used in a manufacturing suite. It was a microcarrier-based process, which means that we didn't have to use the flatware that you saw on the previous slide, so that we could scale up rather than scaling out. And then we created a functional downstream. Our downstream before, which I'll go into in a lot more detail, wasn't kind of functional for any high titer or high volume processes. For our scale up, we used computational fluid dynamics and other type of modeling and our in-house iTransfer engineered design package, essentially ensuring that any tech transfer we do from the client into APC and then APC into VLE is done in a methodical and risk assessed way. And our engineers in the process development were also our engineer in the MSAT space, and our VLE engineer then would have followed kind of throughout the process as well. Rapid tech transfer to GMP was really important. So our scalable single-use equipment, so our equipment that's in our MSAT space is the same equipment that's in our VLE suite, so that we can scale directly from our 50 or 250 litre MSAT space directly into our VLE suite, so that our gap assessment on that tech transfer doesn't have many gaps because the equipment is the exact same. All the single-use manifolds are the same. So everything is very lean and agile in that case. So one other thing I really wanted to mention was kind of our CMC approach at APC and VLE and how we're guided by the ICHQ guidelines. So in terms of process design for APC, we focus heavily on ICHQ8 and the Q8 revised version, looking at pharmaceutical development and how that relates then into our manufacturing house, which is VLE. Overall, APC, VLE, and then our monitoring continuously, our continuous verification, lifecycle management, and tech transfer is all aligned with ICHQ9, so the quality risk management. And then as well, we include ICHQ10, which is our pharmaceutical quality systems, or our PQS, just ensuring that what we're doing is kind of that harmonized approach that's been taken by a lot of big pharmaceutical companies, and that whatever we're doing in our process development labs can be tech transferred either into VLE or out to an external CMO or the client. And that what we're doing is really valuable for the client, the patient, and whatever phase they're at of their drug life cycle. Going on then from ICHQ9 and 10, what is really important to us at APC and VLE is our knowledge transfer and management. So we have what's called iAchieve. So when we have these big projects and we have a client team, we have our VLE, GMP professionals, and then we also have our APC team. We have a lot of stakeholders that are all working on the project together, sometimes in different time zones, in different countries, for example, and we could have different teams for a client that are in different time zones in different countries as well. So for example, in green there, you see we have our innovation team, our process development engineering, quality, scientists, analytical, and then we might also have our GMP engineering team, facilities team, ops team, QC and QA, and then we also have in pink our client. And what's really important for us is bringing everybody together kind of under one roof and having one knowledge source or one knowledge platform, and that is iAchieve. So how we do this is by breaking up our projects down, or breaking down our projects rather, by our design requirements, our objectives, our deliverables. So from the very beginning of the project, we have the end in mind, and that's the same with how we do our process development, is that we always have the end process in mind that we're trying to do from a GMP perspective. And then looking at our deliverables, we have our experiments and our data. And that data is contextualized onto insights. and those insights are then linked to our CMC decisions. This graph just shows kind of what experiments are linked to each other, for example, on the platform. So you can see if you're performing an upstream harvest, how it's linked to your downstream first chromatography unit operation, for example. And you kind of get that thread going through the medicine each step. This is an example of a CMC decision, and then kind of around project planning as well, in terms of how many experiments are done, how much work plan items are completed. So then getting a bit into the science. So our accelerated process development, we have the process that we were given, which is that flatbed or flatware process. And we decided to go through kind of a microcarrier based process instead. We looked at our microcarrier selection, optimizing our growth conditions, selecting our hardware, and then looking at our multiplicity of infection, our time of infection, and then our harvest time through a series of OFAT and DOE experiments. Everything that we were developing, or all the different technologies that we looked at, have to be single use based on the facility that we have at APC and VLE. Sterile because of how big our product is. Our product is 300 nanometers, so it can't be filtered by a 200 nanometer sterile filter, and operationally transferable between the two companies or to an external CMO as well. The process that we ended up getting was hyperstacks going into a 50 liter N-2 and then a 250 liter N stage bioreactor. But what you can see from the comparison of the two processes is that our productivity went from 100 plaque forming units per cell to 600 plaque forming units per cell. So we increased our productivity of our cells by a lot, and we also increased our surface area. So we've more cells, and we also have cells that are more productive. So we've more product and more virus. However, you can see that we've increased our harvest volume from 8 liters to 250 liters. And all of these improvements in terms of more cells, more product, and more volume, you would have to have a very robust and scalable downstream to ensure that you can then purify that material, which unfortunately isn't something that we had at the time. So we increased our harvest volume by 36-fold, as well as increasing the other attributes that I mentioned there. And the downstream that we were kind of tech transferring in was this manual harvest, from where we take off the roller bottles, add water for injection, and then pour off the media, add water for injection, and then we're left with our cells and the water for injection. We go on into batch cell lysis using sonication, DNA digestion with an enzyme, clarification with depth filters, and then a formulation step using UFDF. This wasn't scalable, first of all, just by design, and it couldn't handle very large volumes as well, because everything was manual, and if anyone has used batch sonication, it's usually in a BSC kind of as a sample prep technique. And as well as that, at the stage that the client was at with their clinical trials, the impurity removal and kind of enzyme removal of this process wasn't high enough for the nanograms per dose for HCP, BSA, and host cell DNA that they needed for their clinical trial. So we took the 6 unit operations from our adherent platform is essentially what we added on. And we tested initially kind of a proof of concept feasibility study from our own platform that we have. So this isn't a platform that brings any IP with it. It's just from the kind of vast SME knowledge that we have on site in our downstream scientist team. And then we would have performed OFAT and DOE for optimization. And then we actually tech transferred the formulation step that you see there directly because it worked quite well, and it was very, TFF is a very scalable unit operation. So what this new downstream looked like was our microcarrier removal using a harvest stainer. We then utilized some single-use continuous centrifugation, which is a great unit op. And because it's all, you can get the consumables in gamma irradiated, it was very good for our tech transfer into VLE. Cell lysis, we did a batch to continuous study, and we did a DOE on kind of looking at residence time inside the sonicator, and also looking at our wave energy and our flow rates and everything like that. Then clarification, we looked at different enzyme screenings and different cofactors for these enzymes and different cofactor concentrations. And then we used multimodal chromatography as well. For our scale up, and I think one of the big benefits that we did on this project was compressing our scale up timeline. So we scaled from 3-liter bioreactors to 50 to 250-liter bioreactors on this project. And what we used an abundance of was computational fluid dynamics and also engineering correlations for scaling. For example, using correlations for power per volume, tip speed for KLA, and for using CFD for our mixing times, our strain rate, and also for our columnograph scale. And what we were able to do here was, when we were scaling from that 3-liter to that 50-liter and then to that 250-liter, when we're performing our gap assessment in our tech transfer from our receiving unit to our sending unit to our receiving unit, we are able to use that CFD as one of our studies so that we can be assured of the homogeneity or the hydrodynamics in the vessel. A 3-liter is now matched in the 50 and the 250. So we were able to make really good decisions, and decisions that were backed in kind of engineering and science. And that was especially important to the mixing because we had a microcarrier process. So we had to make sure that our microcarriers were getting suspended, so the just suspended speed of the microcarriers. And at the same time, we have to make sure that we were mixing enough to get a good mass transfer in the bioreactor, but also that we weren't mixing too much that we would break up our microcarriers or in some way get our cells off our microcarriers. So then from our rapid tech transfer from pilot to GMP, so our GMP suite is on the same floor as our MSAT space. They're actually right beside each other, and it would be, everybody sits in the same area as well that would be working on these projects. So from an engineering perspective, it's the same people in the room. There isn't, a big knowledge gap of equipment because it's the same equipment. So in terms of from writing our process description document at the 250-liter scale and going then into our gap assessment and then writing our own VLE process description, they were very similar because of how similar the equipment was. And because we chose our GMP-able single-use tubing and materials and raw materials from the very beginning, that meant that we didn't have a lot of roadblocks from that sense. This process was completely single-use. So I just have, these are images taken online, but we have, aseptic quick manifolds with clamps. We used welders and sealers if we couldn't use our aseptic quicks, kind of as our backup. And then we also would have used some autoclaving as well. And this is the scaled up process. So you'll notice when we compare it to the downstream slides, it's the exact same equipment, all just linearly scalable. So our 250-liter sub is the exact same as we would have had an APC. And then we had all this equipment in our MSAT space, but also this equipment scales very, very nicely. So the unifuge, for example, will be scaled off the sigma factor on the bowl. Our single-use mixer, we would have looked at characterizing those mixers using CFD and seeing can we match the mixing. The harvest stainer, Thermo Fisher had some scaling correlations for these. And then similarly for our ACTA, we'll scale based on our linear velocity and our column size. So it was a direct like for like transfer that we were able to do, which was great. And from that pilot scale that we're talking about on the same equipment in GMP, we had our GMP operators working in our MSAT space along with our scientists, so that we could get an early operator familiarization, for example. We did material flow simulation and de-risking that process then as well, because the spaces are so similar. What was great about this space as well was that we were able to use our batch records from the MSAT space, but also do documentation dry runs. And the kind of biggest speed in all of this was that while we were still developing and running this process in our PD labs, we were actually getting the VLE suite ready. But because we knew what the process was going to be, and because we knew what we were scaling up, because we already had the equipment in the MSAT lab, we were able to get the facility ready for VLE during those pilot campaigns because we knew there was going to be minimal changes. So the key results from this process was that we increased our viral yield by 400%. We had a 30% total processed yield post downstream. And for anyone that's familiar with these oncolytic viruses or these MVAs, they're also called, 30% is quite high in terms of a downstream yield, because that process can't be filtered, or the virus rather can't be filtered, and we have to go through a lot of steps to purify it. Our viral production increased 1,000%, and we were able to decrease the cost per dose by 90% for the client and also, more importantly, for the patient. We increased time to the patient. And as well as that, and what's important kind of from our regulatory perspective and bringing it all back to the safety of the patient and the efficacy of our drug, we impure our impurity profile was either matched upon or improved, and we made a process that was robust and scalable so that we could meet demand for patients while making sure that it didn't cost a significant amount of money and that our supply chain was reliable. Some challenges and future learnings for this. Scalability challenges. So although we had a very positive scale up experience, there were still some things maybe that we missed that when you're doing something at 3 liters versus 250 liters, there's always going to be differences that you find. For example, cell settling in the 3 liter to do kind of a media exchange is a very different process at 250 liters, and kind of anticipating the time changes and how that affects the cells and the stability of our process. That was very important. And then also looking at single-use manifolds. So single-use manifolds have been around for a while now, and single-use technology as well, but still aren't very well understood in industry as a whole. So as I said before, APC and VLE are complete single-use facilities in terms of our large molecule capabilities. But what was really important kind of for us was around our labeling of these manifolds and the qualification of these manifolds in terms of getting them from suppliers in time and beating those supplier lead times, because this project was done during the COVID pandemic and getting single-use bags and single-use manifolds was very, very difficult. So standardization there, in my opinion, is absolutely key to make sure that you're getting from the same bucket and you can do quite large orders from suppliers and kind of get priority then. A big thing as well for kind of ATMP and single-use technology processes that are coming through in the industry nowadays is that the operator completely owns the process. So, maybe in our more traditional stainless steel kind of reactors that we have, we can do the automation on the valves, pneumatic valves, for example, on our computer screens. However, when we go into these type of processes, it is the operator that's connecting manifold A to manifold B or tube A to tube B using aseptic quick connections or using welding, and then they'll also be sealing those tubes as well. And what's really important about that is it's not just following a batch record, it's making sure that they know which tube on the bag are they going to take, because those things can actually end up taking a really long time. If you're looking at harvesting a bioreactor, if you were the engineer that designed the tech transfer and did the gap assessment, you know off the top of your head where that tube is. But it's really important for the operators to know what they're actually doing and what tubes they're connecting to what, and what is C-flex and what is silicone, and why it's important not to pump on one, but it is important to pump on the other. And that's kind of where it comes into the power of workshops and why it's so important to have your kind of walk the batch and bring your operator along the process with you, because they're the ones at the end of the day that own the process. But yeah, that's everything from me. Thank you so much for taking the time to listen. And that has been our webinar on the process development of an ATMP and kind of bringing that to scale up and GMP. Thanks so much.